And our first speaker in the afternoon session is not new to Dartmouth, to Thayer, and he is Drew Endy. And Drew is a Dartmouth alumnus, and he knows this place better than me and better than many of us here. So Drew received his PhD in biotechnology and biochemical engineering from Dartmouth in 1998. And in the very Dartmouth spirit of being a great teacher as well as a fantastic scholar, Drew developed the world's very first fabulous genetic engineering teaching lab in the new bioengineering program at Stanford. And previously, he did the same thing for another wonderful school on the East Coast, MIT, and started a biological engineering major at MIT. So his Stanford research team developed genetically encoded computers and redesigned genomes. And he co-founded quite a few remarkable organizations. So this includes BioBricks Foundation as a public benefit charity supporting free-to-use standards and technology that enable engineering of biology. He also chaired a variety of activities of national, international significance. For example, he co-organized International Genetically Engineered Machines, or IGEM, competition, the BioFab International Open Facility Advancing Biotechnology, and Gene9 Incorporated. He also serves quite a few very important committees. So this includes the U.S. Committee on Science, Technology, and the Law, and is now a new voting member of the U.S. National Science Advisory Board for Biosecurity. And Drew also did fantastically well in creating a few centers. So he, the Esquire, I think that's a very important journal, named Endy one of the 75 most influential people of the 21st century. And he offered quite a bit of advice during the student luncheon just finished minutes ago. And also worth mentioning that Drew's wife, Professor Christina Smoke, is also a leading scholar in bioengineering and also a faculty member at Stanford. So an exciting field like bioengineering, biomedical engineering really can draw family attention. And with that, I will turn the podium to Drew. Let's welcome Drew.
Happy 150. Totally amazing. This is my last slide as well as the first slide, and I just let you know that because a free copy of the slides is at that URL, slideshare.net/my name, and go for it. I'm going to talk about frontiers in translational medicine, looking at biology from the inside out, right? So far today, we've started with hardware connecting with the biology, and then we've moved into the biology, and I'm just going to go totally into the biology and think about bringing it back to the engineering and what that might mean and related to translational medicine. I love this film here because it reminds us that our planet is already dominated by a type of material, living matter, that's beautiful and esoteric and mysterious and sacred. And we're now at a point in our civilization's development where we're getting better and better at tinkering with it as we understand it. I want to open just as a type of set of on-ramps with examples of how we've historically approached engineering living matter. Most are familiar with the objects shown here, but perhaps not in the form demonstrated. These are the natural plants from which food crops we're more familiar with are derived. And the process by which these turn into domesticated plants that we consume is well documented and so on and so forth. I'll illustrate it with animals on the following slide.
We really enjoy the ability to take the gene pool and use it like paints. It's our art. This is my art. I made this beautiful dog that I enjoy. I made her. I chose her sire and her dam. I chose several generations to make this beautiful dog. I'm very proud of.
Selection for trait, breeding, continued selection. You shape biology literally by that algorithmic process. We also, as a civilization, shape biology by shaping the context within which biology develops or operates, for better and worse. Here, the deforestation of jungle and forest to make room for oil palm plantations to make bulk chemical products effectively. massive impacts. About a human generation ago, not a surprise, we started engineering biology by changing the DNA directly, recombinant DNA, patented circa 1980 and brought to market. We can quantify how it's gone to market over that first couple decades. So this is a representation, the most recent representation you can get. It's already 5 years old. It's the genetically engineered domestic product in the United States. It's about 3% of our domestic economy. These are things sold in the marketplace that are made by genetic engineering. And it's coarse-grained into 3 big slices of the pie: biologics or drugs, which a lot of people think about and is the focus of our symposium today, industrial enzymes for manufacturing, and then food. Year-on-year growth looks pretty good, and most of it is still ahead of us in terms of economic opportunity, I would say. Examples of these areas of activity include taking a microbe and reprogramming its genetic code so that it'll take in a commodity feedstock like sugar and make an oil or an industrial intermediate chemical, shown here, done by a company, Amyris in Emeryville. You mentioned my wife in the introduction. Her team, just under 2 years ago, were the first to demonstrate that you could take yeast, brewer's yeast, and add 30 enzymes from 6 different organisms into that creature and get it to make hydrocodone, morphine, opiates, things that are typically sourced from the poppy and used to treat pain with lots of problems. They now can do that via a brewing process. So you don't need to go to the poppy fields in Tasmania and get the poppy straw and extract the stuff from that. You could feed sugar to a fermenter and out comes this stuff. That immediately goes to market in the form of a company. What's the purpose of that company? Is it to displace poppies? I hope not. I hope the purpose of the company is to demonstrate engineering mastery around the 30 enzyme biochemistry to get to analogs that don't have respiratory depression, constipation, and addiction, that maybe get to a material supply chain that serves over the 5 billion people on Earth who don't have any access to these medicines, but you might be able to get to them, provide to them if you could brew it locally. We'll see. Dennis's work with the papaya in Hawaii, making a virus-resistant form of this food crop to save the industry there. At the time it was done, it wasn't that controversial, but it's controversial now because it's genetically modified food. So now we're here. So the examples I've given you sort of catch us up to today. And I took the invitation, which I'm very grateful for, quite literally. We're supposed to talk about diagnosis, treatment, and prevention of human diseases at the levels noted, but also talk about recent progress on the frontiers and the entrepreneurial process and next generation engineering education. So I'm going to reflect on what I do most of the time, which is related to the bottom half of this, I would say, and link it back to the topic of the symposium. It's interesting that about a year and a half ago, the engineering school at Stanford published our first ever set of strategic planning documents. They're free online. And they took the form of 10 questions. The questions aren't meant to be rhetorical. They're things we believe we can go do. One of the 10 questions was, how good can we get at engineering biology or living matter? And we believe, to a first approximation, that we can get incredibly good at that. And I'll give you some evidence for why we feel that way. I'll simply note that the second bullet point, and the list continues, open new frontiers in medicine wherein engineered cells sense, diagnose, prevent, and treat diseases in place. Literally, almost perfect homology to the topic here before us today. Why do we think we could pull that off? Well, if you look back in time, going to, say, 2003, you can document what we were struggling with and talking about. Here, for example, is the briefing to the director of DARPA I presented in October of 2003 on the topic of synthetic biology. The charge of this activity was to say, biology is incredibly important. Could we get a lot better at engineering living matter? If so, what moves would one make to systematically advance the engineering of living matter? And we took 18 months of time and brought 50 people together from both engineering and biology and tried to write down what the best ideas were. There were 3 ideas that came out of this, and if you read the doc, you can see it. They were informed by things like the advances in electronics, but we weren't trying to merely recapitulate the history of developing electronics, just adapt lessons from other fields. The ideas mostly had to do with how you would take the core of an engineering project, where once you've figured out what you want to do, you have to design a system, you have to prototype it, build it, test it, see how it does, and when it doesn't work, you've got to do that again and again and again until you have something that works. How would you make that better, that cycle, for living matter? And the ideas we wrote down in '03 were separate design and fab by using a tool called DNA synthesis and getting much, much better at building DNA so that some people would become expert designers of DNA, but they would pay somebody else to build the DNA. Develop standards for coordinating labor so you could reuse materials and measurements, and manage the complexity of biology by adapting concepts of abstraction from electronics and programming into living matter at the molecular and cellular scale. These ideas were very poorly received by both the government and the research community. In 2003, when one proposes to get better at printing DNA, the director of DARPA interprets that as people will use this to print pathogens. We've just had a bioterror event. I'll actually reduce funding for getting better at building DNA. There's no such thing as a standard biological part because everything in biology is uniquely context sensitive and you won't get reliable reuse of sophisticated genetic functions and so on and so forth. Nevertheless, the world has played out in ways that have practically overcome that initial rejection. So let me give you the quick run through of that, and then I'll turn to the translational medicine aspect of my talk. Shown in the bottles here on the top of the slide are the bases of DNA, A, T, C, and G, that comprise genetic material that encode the behavior of living systems. The bases in these bottles are not derived from any existing organism. They're made from scratch. Each bottle costs about $250 and has about 10 grams of each of the bases. You can connect these bottles into a machine. This is a decade-old machine, so it's out of date, but it still works. And this machine is called a DNA synthesizer. You can think of it like a musical synthesizer, but instead of having 88 keys on a piano, you have 4 keys. And if you press the A key, you get the base A played on your synthesizer, and T, C, and G. And so depending on the sequence of information you input into this machine, the machine will dispense the chemicals in the right order to make from scratch the molecule of DNA that you specify. Okay? So this allows you to go from abstract information to physical genetic material, and if you can operate that inside a cell, you then get the so-encoded behavior to the extent you know how to program DNA, and you have a rule or framework for composing information into DNA. When I first started teaching, the discounted prices on building gene-length DNA was $4 a base, and that was because the CEO of Blue Heron was an MIT graduate, and he gave us a discount on the cost. This year, to first approximation, we're about 4 cents a base. So we've seen 100x cost down. I've been part of 2 companies working on this. The second was just acquired, but not for a lot of money. It turns out to be really tricky to run a business when the price you can charge for your product drops by a factor of 100 over just over a decade. That means you have to do 100x more volume to make the same amount of money, but you're doing 100 times as much work inside the organization. So we have succeeded technically in changing the economics, but we haven't yet figured out how to make a lot of money with DNA printing. And I mention that because it's really important. DNA is you can think of it as an industrial polymer of our time and for the future. And how we get a strategic plan to keep getting better at this is just of massive importance. Does this keep going? I don't know. Physically, it seems like we should be able to pull this off from a technology perspective, but how to organize people to get it done or companies to get it done hasn't been solved. I want to ponder this type of transition, though, as it relates to the topic of the symposium, because it's really strange and tricky to think about how you should position yourself strategically for when things are changing at a fundamental level in dynamic ways. So let me give a side example, and I'll bring it back to biology in a second. I presume most are familiar with Pixar as an entertainment company. Pixar is famous for making computer-animated feature-length films, and they seem like a great success. But what's important to know about the history of Pixar is what you might know them for was not their first experience. In fact, if you read the interviews with the co-founders of Pixar, here's one of my favorites, effectively they failed twice before they made the movie Toy Story. And the first time they failed had to do with getting so excited about making a computer-animated movie that they just went ahead with the project and then discovered that it was too expensive to render the animation because the compute cycles were too expensive. Then they said that was a bummer, let's catch our breath and come back and do it again. But because they had failed the first time, they knew enough to run the economic calculation before they finalized the deal, and they determined that they were still 5 years too early from an economic perspective to afford to render the full-length feature animation. But now they had 2 data points. They had self-taught themselves regarding Moore's law and the pace by which compute was improving, so they could estimate that in 5 years into the future, they should do what becomes Toy Story, right? So take that for what it is, but it's a nice, accessible example, in my opinion, of how when you have a fundamental technology trend that's moving out, where you might think about first learning what the trend is, 2, trying to extrapolate and be strategic about when to position a business move. So let me bring this back then to the world of DNA and eventually medicine. Here's the first question on the final exam from Introduction to Bioengineering at Stanford back in June 15. This is a course I teach with a colleague, Jan Lippert. We have about 140 undergraduates now in that course. We're very generous in our grading. If you put your name on the exam, you get 2 points out of 100. But take a look at the first question. So over the past period of time, cost of building gene-length DNA has dropped a factor of 100, about a 2-fold drop every other year. Meanwhile, we've got another trend that's not so great. The cost of attending Stanford doubles every 15 years. We could debate and discuss why the trend for that is not in the opposite direction, but that's what it's been. So presume a human genome is this long, a couple gigabases, and these 2 trends continue. Stanford tuition increases over time, doubling time of 15 years. DNA build costs drop in 2 every other year. At what point in the future will the cost of attending Stanford for 1 year equal the cost of printing the DNA encoding a human? Not just editing the DNA, but like full-on print the whole genome for a human. Now we'll grade this on the math. Can you solve the intercept of those 2 equations? Anyone want to take a quick guess of what the time in the future is?
5 years.
5 years. Not bad. It's about 19 years from now, we estimate. So 2036, I guess that would be. So we'll grade that one on the math. Part B, we'll just grade on, can you come up with a position and explain it? Do you and your future partner, if you want to have kids, do you start saving up money for college so they can go to Dartmouth or Stanford or wherever? Or do you save up money so you can print their genomes with whatever changes you wish? This question, again, we don't take a position on what the answer should be. It's just, can you reason your position? Splits the class. About 60% are morally opposed to the idea of printing human lineages. That's not what it means to be a parent, so on and so forth. 40% are, yep, going to do it. Cost basis of education will change. Khan Academy 10.0 will exist, or something like that. And meanwhile, if there's any advantage to be secured to my offspring via this new approach, I want to be prepared to enable them to realize that. So that was DNA synthesis. And again, it's a very fragile but precious technology that's moving out in an interesting way. How you try and position yourself to navigate that in the future, I offer you one exercise. I'd encourage you to think about it. DNA is in the middle of all of biology, so it's important. The 2 other concepts from 2003 were abstraction and standard. So let me take those quickly. Here's a made-up abstraction hierarchy for engineering integrated genetic systems. Let's say, for example, I would like to program cells to count to 256. So I could track cell division events and use that to determine the age of an individual cell or how it develops throughout the realization of an animal from an egg. And maybe I'm going to have an asynchronous counter like shown on the top here where I'm flipping DNA back and forth, and eventually I could count to 2 to the 8th because I have 8 digital registers in some system. What DNA base pair would I start with to implement such a thing? An A, a T, a C, or a G? And that's an incredibly impossible question to answer systematically because it requires being able to, in your mind, go from this high-level description of what you want, say an 8-bit counter, down to the primary machine language, if you will, of the cell, the basis of DNA. And so instead we have this notion that maybe we should focus on devices, simpler things like flipping DNA back and forth. We're going to implement these devices from actual molecules that carry out the biochemistry and genetics. And only then would we eventually compile down the DNA. People would become specialists at each layer with defined interfaces, allowing for limited exchange of information across the layers in the hierarchy. This is how we're going to attempt to manage complexity in biology. This is very controversial. And note, by the way, again, it's all made up. This is a human-engineered construct. This may be the right way to go about it. You might have another way to go about it. Maybe we shouldn't be going about this. So I'm just going to admit to that. So we worked on that for quite a while. I'll give you an example. We decided we wanted to implement a 1-bit digital register based on flipping an engineered sequence of DNA in the middle of a chromosome. And if we could flip it and flip it back, that's 1 bit of information. We could use that to control in a digital fashion, turning genes on and off. Although this example is digital, I don't mean to take a position on whether biology is digital or analog. It's both. This just happens to be a digital example. And we did it 5 years ago. But what I want to emphasize is it took us 3 years to make a 1-bit digital register on a chromosome. And it took us 3 years because we didn't know how to design the DNA to do it. And so we had to take 700 attempts, kicks on goal, so to speak, different sequences of DNA to do something that ultimately isn't that complicated. We're just expressing 3 genes that have to have a particular dynamical trajectory within the state space of the inside of a cell. And if we get that right, we could do this flipping and flipping back. And Jerome, now in Montpellier, France, running his own team, was the person who led the work. He thought he was coming into a different context. As a biologist coming into an engineering school, he thought he'd be able to get standard biological parts that would work off the shelf, that would snap together, just like in electronics and a breadboard and TTL, and he'd have his thing that would work approximately the first time, right, or approximately the first week. But no, it wasn't like that at all. Instead, it took him 3 years, and when I presented this one time at the law school, the dean of the law school asked, how long would you have kept going if it didn't work after 3 years? And the answer was, until we ran out of money. So anyway, there's a couple other lessons baked into this. So when we first started approaching this type of work, implementing genetic logic and data storage inside cells, we were dominated in our thinking by Shannon and his work to adapt earlier thinking into electrical relays and then what we're very well familiar with in terms of electronic architecture. The puzzle there, or the baggage associated with that work, which is obviously awesome, is it pre-constrains how you think about the architecture of the devices. And biology turns out to be different than electronics in its fundamental physics, and so you can do things with different device-level architecture, which I'll get to in a second. We didn't understand that until we went back in time and studied the work of Boole, where when you look where logic comes from, in my assessment, it comes from an analysis of language, which comes from an experience of the universe and the world and how we practically interact with the things we observe and try and do. And there's no, in other words, when you look at language, there's no physical specification for how you should implement logical functions in any particular type of material. Just adapt the functions into the material as the material best allows. Let me give you an example of this. So we had just gotten good over a period of 3 years at flipping DNA. We could then set out to flip arbitrary sequences of DNA. The thing we're going to flip here is the equivalent of a genetic diode or a check valve. It's an asymmetric transcription terminator, this T-shaped object in the middle of this schema. When DNA is read out, that's carried out by the flux of an enzyme, RNA polymerase, transcribing the DNA. If the RNA polymerase hits that check valve, it'll abort the reading out of the DNA. So if I have something being read from left to right, when the polymerase hits that thing, it stops. Now, if I can flip the DNA, because it's an asymmetric check valve, the polymerase will pass through and read from left to right. If I flip the DNA back, it'll stop again. So this turns out to be the equivalent of an exclusive OR logic operation. I have 2 different control signals, depending on what I wish to have control, come in and flip the check valve or flip it back. And if one or the other signal is present, it's permissive, and I'll have a high pass-through. But if both signals are present, it'll block again, right? If you were working on something like this in electrical engineering, you might think of implementing an exclusive OR function by having 4 cross-wired NAND gates in 3 layers. But because in biology we can use the spontaneous mixing of molecules to land on the same spot on a DNA wire, we can, through a first approximation, integrate multiple control signals into the same spot. So it'd be like having multiple wires coming into the same transistor, controlling what's going on in the transistor. As a result, you can just realize a different device architecture that's simpler. Let me show you what this looks like inside cells. Those are cells growing and dividing, just simple bacterial cells. The blue signal in the middle is the control signal that is indicating how much information, the strength of the signal needed to flip the DNA, and then the signal on the right is showing the change in output in the gate. So not only were we, and you should just be able to see the right is brighter than the middle, which is demonstrating amplification. This is the first time anybody's got genetic amplification working in this way. The amplification is about 20-fold. So the change in the gate signal is about 20x the change in the control signal needed to flip the gate. Fine. One of the reasons we were doing this was to get computing working in places where existing computing modalities won't work. How do I implement logic not on top of my cells and transducing information into cells or out from cells, but inside every cell, right? And if I can implement computing in a new place, I'm not competing with silicon or other computing modalities. I'm just adding value because now I have logic and memory under my control in a new type of material, in this case, inside living cells. I was surprised how quickly this translated, and coming here yesterday, I remembered we had a paper just in 2015 on the cover of Science Translational Medicine where people were very quickly able to take these DNA flippers and drive them with biosensors that would be responsive to analytes in serum or signals in urine, could move these gates from the organisms we had developed them in, E. coli, into other microbes that are more relevant for operating inside fluids associated with humans. And in this particular case, the idea was to simply have wetware operating as a diagnostic platform that could provide an affordable diagnostic platform that you could deploy so that people could determine whether or not they had early indications of diabetes or other diseases. In the same issue of this, I'll just mention Jeff Hasty from San Diego was doing similar work, but with cancer detection. Right? So to the extent you can transduce information into biology and then map that information into a DNA data register and then use that to either go onto a sequencing platform and read it out or just use that to reprogram the biology to make an obviously observable signal, the wetware itself becomes a pretty compelling diagnostic platform. You could then easily imagine, as perhaps we'll hear about, moving that forward into treatment as well. I don't want to belabor this, but there's something that we didn't put in that paper back in 2013 around the logic gates. It turns out, unlike the work we had published in 2012, it took 700 attempts to get it to work. Every DNA design in our 2013 paper worked the first time. It only took about 6 weeks to do that work, so it's fast for biology, slow for electronics, but fast for biology. And how come that was the case? And it turns out that the reason it was the case is over a period of about a decade, we had finally figured out in a very limited set of examples how to make reliably composable and therefore reusable standard genetic elements. And this little movie here shows just one example where if you want to initiate the synthesis of a protein, you need a particular control sequence on the messenger RNA. Let's say you have a really good control sequence. That control sequence doesn't really know what you want it to do. It's just a molecule. And so when you attach it in some arbitrary way to your favorite gene, it could be that it'll tie itself up in a knot with the coding sequence of that gene. And if that knot gets formed off to the right, it won't work at all. It'll be tied up and occupied. It turns out that the solution to getting reliable physical composition is to use the biology itself as an active process to drive reliable behavior. So the answer in this case was if you want to reliably express a gene, regardless of what the sequence of the gene is, to have a much more complicated head unit, if you will, that initiates the process of protein synthesis via a ribosome, recruits the ribosome to a leader peptide. The ribosome then makes a very small molecule, only about 11 amino acids, but the intrinsic helicase activity of the ribosome unwinds any secondary structure or not that's disrupting the behavior you wish, the ribosome reinitiates and in a very reliable fashion that you can quantify goes and reads out, okay, yada, 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 10 years, we showed that you could get reliable functional composition for certain types of genetic elements that otherwise in the literature people were declaring it was impossible to do. A way of showing that in a heat map form is on the left, a panel from strong to weak of different protein synthesis sequences. The columns are all different genes. The color indicates the amount of protein made. You can see it's a salt and pepper board, not very reliable composition. We use the more complicated physical architecture indicated in that movie, and you get much more reliable composition. I can quantify this. I can say, give me an arbitrary coding sequence for a protein. I can get that protein synthesized to within a factor of 2 target concentration with 93% reliability or 7% failure rate. Having shipped this product back in 2013, although it helped us with that one demonstration around logic and got Jerome's stuff working much more reliably, we also discovered that there really isn't a market yet for standard biological parts that work reliably, because very few people know how to design or just come up with a description of a system that specifies with precision how much of certain things are needed to get the right behavior. Okay, now, however, this keeps going, and so I just want to highlight some work coming out of MIT, a good colleague, Tim Liu, paper published very recently extending this sort of work. What's really neat in the field of synthetic biology right now is how much reuse and reliable reuse of materials is starting to occur. So if you pull the acknowledgments section of this paper, I knew nothing about this project. We weren't competing. They're just off to the races. And it turns out that the things they're using to flip the DNA in their work are all the things we developed in our labs back at Stanford and we're sharing freely through an open source platform for reuse of reliable materials in genetics. Okay, so now I'm going to pivot and move a little bit into education, and this will then quickly return back to translational medicine. Bill Joy, well known in computer science and perhaps to all, is famous for apparently making the following remark. As shown here, I'll make the shorter form of the remark. Most people work for other people. No matter who you are, most people work for other people. And it's not an arbitrary declaration. It's sort of a lead-in to a hidden question. So what's your strategy for that reality? If we're all operating in a world where most people work for other people, what do we do? Most engineers don't come to Thayer. What's Thayer's strategy for that reality? If you're Larry Page running Alphabet, you're only employing directly 1 in 10,000 people on Earth. What do you do about the other, set of people? Now, in Sun, as I understand it at the time, this contributed to what becomes Java Virtual Machine and the Java programming languages that allowed for computer programmers to share code regardless of the hardware operating system lock-in that was established. And indirectly, then everybody gets to work for Bill. But much more importantly, everybody developing in Java has a chance of working for everybody else developing in Java. Right? I think that's the fundamental hack that I'm intrigued by reflecting on this, regardless of what he meant by it and regardless of whether he said it or not. I just think it's important. So why is that really important for translational medicine and bioengineering? Most of biology isn't discovered yet by a long shot. Most of the genetic gizmos I'm going to want to make the stuff somebody else is going to tune up and get working really well. Most of the problems that could be addressed with biology, I know nothing about. Most of the biomanufacturing capacity on Earth is under the control of other people, and so on and so on and so forth. So I think Joy's law, as it applies much more in biology than in any other space of technology we've encountered. I would, back at MIT, get this well-intentioned but ultimately limiting question all the time from my more senior colleagues and my parents. What disease are you working on? What disease are you trying to cure? And it's a good question, but eventually I realized I couldn't choose because there's so many things that could be done, and I became much more interested in thinking about how to make it easier for people to do such work such that it would all get solved not at some arbitrary point in the future, but in a much shorter period of time. So that combined with the fact that the government didn't like any of this from a risk perspective meant we took it to the students and tried to see what would happen. So here are the first set of students we had in '03, and approximately it's become this. Randy Redberg and Megan Lizarazo have led the development of this amazing thing called iGEM, the International Genetically Engineered Machines Competition. It's a genetic engineering Olympics. Think of it like FIRST Robotics, but for wetware, that happens each summer. Last year was about 6,000 students across 300 schools, and about 3,000 of them come to Boston around Halloween every year at the Heinz Convention Center. It's well outgrown the campus. What's happening in this activity is at the start of the summer, the iGEM headquarters gives all the teams a kit of parts. The part kits, got a couple thousand DNA parts in it. Not all of them work. A few of them have been measured. That's way better than nothing. And teams do whatever work they want to do. They come back to this competition. To get a gold medal, you have to give back at least one part from your project that's new, and that creates a ratcheting that lets the part collection grow over time, and so on and so forth. And I just encourage you to check it out, and I dearly hope there's a team here at Thayer, or will be, and I'm happy to help. In any case, why might that be interesting, and why might that be interesting in the world of translational medicine? So on this website, and remember, the slides are up. I'll give you the URL where the slides are so you don't have to jot any of this down. On this website, you can see one-sentence summaries of every iGEM project starting in 2008. All right, so here are all the projects in 2008. About a third of the way down. Dr. Coley, a microbe to release drugs in the human body and then self-destruct when the disease is gone. These are teenagers who are encountering biotechnology for the first time, coming up with what they think bio could do, and then attempting to prototype that project. Oral hygiene yogurt, 3 from the bottom, and so on and so forth. So some of these projects are approaching diagnostics and treatment, and so on and so forth. Let me cut to 2016. All right, so here are the projects from 2016. It's approximately more of the same. So you're going to see energy, you're going to see environment, you're going to see health, you're going to see education as sort of some of the major categories. Let me give you an example of one as it relates to health. So this was a project from 2011 from the University of Washington in Seattle, and they wanted to treat gluten intolerance. And they thought that an approach for doing this would be to engineer an enzyme that do a better job of basically dealing with this in the context of the gut. And if you go to the registry of parts for the competition, you can see BioBrick Alpha K590087, designed by Sydney et al., was contributed to this. They called it Kumamax, mutilated Kumamolysin. Okay. See, note these people don't work for me. Note these people know a whole bunch of stuff I know approximately nothing about. I can learn by going to their wiki. And they're sharing this knowledge with everybody in a world-readable fashion, and so on and so forth. Ingrid was part of that team. And last month she just raised 35 megabucks to take her company into trials, bringing this engineered protein that has since been further approved and looks like it's got a good chance of working for treating this disease. All right. So now imagine this happening to the tune of 6,000 people a year. All right. The student in the front row center from 2003 is now the president of Ginkgo Bioworks, employing 100, going on 150 people down in Boston. And that's kind of interesting. Why is this happening? And I want to be really careful about something. So Ingrid, who helped get the first UW iGEM team going, didn't go immediately from the iGEM Jamboree to her $35 million round, right? She spent 5 years of follow-on work, very carefully upgrading and improving that, and then, suddenly there's success. But you can read her story here, and it's just the narrative of how biology, the wetware itself, is a compelling technology, and if you can get onto that journey of engineering and partnering with it to solve problems, there's a lot of low-hanging fruit. And I'm not saying anything that's surprising here. There's a lot of local success that's off the charts. But I think there's a question of how we scale that success and enable that to play out in ways beyond what we might easily imagine. I'll point you to another example of this. So Jenny Rook here is now at 5 Prime Ventures, but at the time she was running a program for the Gates Foundation, a global grand challenge program for the Gates Foundation. They say, we'll give money to people who want to apply synthetic biology to global health problems. And that triggers 700 people apply. And if you read this paper, you can see the summary of the projects that they funded. diagnostics, manufacturing, pathogen detection, probiotic-based oral vaccine delivery system, plant-based vaccine development. So it's manufacturing, therapeutics, diagnostics, sensors, and so on and so forth. I don't think there's any category here which is surprising. It fits right in the themes that have been identified. And so maybe there's a gap we haven't encountered yet in the application space of bio, but otherwise, off to the races. One of the things you encounter when you look into this is a rethinking of how our civilization operates. So coming out of industrialization and centralization and the 19th century metaphors that we inherit, you now encounter biology, and as biology has been industrialized, we move living matter into these centralized manufacturing capacities. I'm not sure that makes sense all the time. So a really neat iGEM project from 15 out of Paris looked in India at the fermented breakfast breads that are made and asked, what is the nutritional content of these breakfast breads? The fermentation of the breads is an opportunity to change the biology operating inside the bread and perhaps address micronutrients was their primary issue, vitamins or iron bioavailability. How do they go after this project? Well, they go there and meet with people who are actually practicing the craft of making breads and seeing what they might want and how they might feel about some crazy idea some teenagers in Paris would otherwise naively impose on somebody and get some feedback. Have some empathy, in other words, for some bottom-up biotechnology. And, just if you go again onto the team wiki, you can see a quick example for how they're going after provisioning vitamin B12 in this bread manufacturing process. The solution here is not to rejigger the genetic code of any given microbe, but to bring into the consortium of microbes that are used in the bread making process a microbe that naturally produces vitamin B12, right? So it's not always that you have to go in and, do the genetic engineering. Locally, we've been experimenting with this a little bit as well. So we have our formal undergraduate curriculum at Stanford, but over last year, we've decided to take the entire first floor teaching lab space and turn it into a maker space for wetware. And we call it Biome, Biological Interdisciplinary Open Maker Environment. We have, to a first approximation, no idea what we're doing, other than to say that there's a lot of people who would like to get access to biology technology, but have very high barriers to getting into the laboratory. And to the extent that we can figure out how to remove some of those barriers, all of a sudden we might see a lot more stuff happening with living materials. When we wrote that strategic plan for the school, how good can we get at engineering living matter, that didn't point back to me or to the bioengineering department. That pointed to the entire school and effectively said, all hands on deck. We need the computer scientists, the people in aeroastro, the people in material science, the people in management science to think about this engineering challenge. And so now we're reacting to this locally to see if we can pull this off. I'm curious, the maker spaces here are unbelievable off the charts. And I already sent a photo of the vending machine for tools back to the building manager because we definitely need stuff like that. We had a big flood in the building over the winter break because somebody had a clamp on a condensation column that fell off and then the cold water supply went all over the place. There's multiple problems there, but never mind. If we only had tools available, though, it probably would have been avoided. But there's a problem here as well. The problem is, like Stanford does a really good job, as does Dartmouth, of rejecting students. And we take pride in the doing of that. And it's really backwards. 19 out of 20 people or 9 out of 10 people are turned away on a planet where the college-age population is expected to increase by approximately 1 billion people between now and 2025. So how could we possibly think this is a good idea? If you go back and read the charter of the university, Mrs. Stanford wrote to have an influence in behalf of humanity, not on behalf of humanity. We're having a jolly old time in Palo Alto, but out there in the world doing the work of making the world better. And so even though it's a neat idea, and I hope you do something like this here if you haven't already, this isn't going to scale. So now let's return to iGEM. And from one perspective, going from 16 students a year to 6,000 students a year, you could say that's pretty good. And note, this is not for just an online digital course. This is a physical activity with intensive labor involved. So that's massive scaling. But 6,000 people a year, if you did iGEM last year, you were less than 1 in a million people on Earth. That's approximately the literacy rate for the frontiers of genetic engineering. Does that seem like the right? Maybe some people would think that's way too many people doing genetic engineering. But I'm going to say, I read on Quora that there are 20 million people on Earth who know how to program computers, 10 million who are professionals and 10 million who are hobbyists or hackers. I think the scale of bioprogramming literacy should be about 1,000 times bigger than iGEM. How do I get to not 1,000 more people, but 1,000-fold more people? Could we do something like that? And so about 2 years ago, we got a $4 million grant from the Helmsley Trust. And the idea was to implement the equivalent of a sneaker net, a peer-to-peer distributed network for scalable exchange of functional biomaterials. What I mean by this is if you look at how the iGEM competition works, teams contribute parts at the end of their successful projects, and then next year's teams get those parts at the start. And that's scaling really effectively. They're shipping 10x more parts than any company I know at a price of $4 a part. But the problem is it only happens once a year and it goes through a centralized node. And so that doesn't work. And getting everybody together in Boston on one weekend every year is also very expensive in terms of calendar management. So how do I get distributed peer-to-peer asynchronous practice? And we think the way to do that is to create a type of infrastructure that doesn't exist enabling this. It includes sort of 4 dimensions of work. We have software developers creating a new type of database and interface for inventory management and query and replace. We have new hardware that's in the works. We think we can replace -80 freezers that use 20 kilowatt hours of energy a day to keep stuff in this big box like you have in the hallways out here with a 10-centimeter cube that operates off a solar panel if you need to. We have lawyers working on upgrades to the material transfer agreements that are state-of-the-art as of 1997 in the United States and don't allow for redistribution into industry from academia, and just let's do better than that, and so on and so forth. So if you go to this website, I won't belabor it, we're launching this over the next 6 months and have just basically an open network for allowing a lot more people to give and get functional biomaterials. And we mean this to obviously connect academia, but without barriers, go in and out of industry as well. Another thing I've been caught up in is we've been invaded in Palo Alto by NIST. The National Institutes of Standards and Technology sent a team of 10 scientists as homesteaders from Gaithersburg, Maryland, and they've moved and now live in California and are in our building and in the genetics department as well. And we're prototyping what we now call JIMBY. I'm the co-director of this thing. It's the Joint Initiative for Metrology and Biology. One of our inspirations and points of departure is JILA in Boulder, Colorado. Before it just became JILA, it was the Joint Institute for Laboratory Astrophysics, which was NIST's investment in making very deep fundamental measurements around the physics of electronics and things related to time and so on and so forth. But now we're looking to making a similar sort of investment over the next 50 years for living materials. So I'll just point you to that website, and we're at about 10 people a year. 10 people right now. We're looking to grow to about 200 people over the next decade. What are some of the things that we're doing inside JIMBY? So some of the things we're doing are just shipping metrology products for biomedicine and for biomanufacturing. So JIMBY scientists are responsible for something called the genome in a bottle. If you wanted to have your genome sequenced and you needed the sequencer to be calibrated against a reference genome, where do you get that reference genome from? What's the metrological object that qualifies your genome sequencer? Well, it comes from JIMBY. It comes from the Genome in a Bottle project that NIST has operated and is now inside JIMBY. But in addition to the metrological products, we're also going deep in terms of the biomeasurements and trying to gain mastery over engineering biology, not just at a couple enzymes or a couple dozen enzymes at a time, but the whole gosh darn cell. So here's a paper that's coming out tomorrow that's just a neat example of a fundamental measurement. In the genetic code, you've got these different letters, bases of DNA. They get transcribed to RNA, and then the RNA is translated to protein, and there's a coding scheme. And the translation from RNA to protein uses a triplet code, and there's a particular code that initiates that process. AUG is called the start codon. If you have those 3 letters on RNA, that will cause the ribosome to start making a protein. But there's 64 triplet codons, and it turns out nobody had systematically measured protein synthesis initiation from all 64, which, for reasons I won't belabor, for reasons having to do with we tried to disrupt start codons and kept getting proteins made no matter how we broke them. And we're like, what the heck's going on here? So then we just said, let's go deep and measure how all these things behave. The short and the long of it is about 45 of the 64 codons initiate protein synthesis above detectable background, and 17 of the 64 codons make proteins at rates that are higher than the known error rates of the ribosome. So as of tomorrow, as of now, I'm going to say, there's over a dozen codons that look to be initiating protein synthesis in ways that are non-trivial. The reason this matters is when you think about a cell as an object and say, well, it's just a couple tens of billions of atoms, couldn't I possibly come to understand all of them and master entire organisms? And then you'd look at things like, well, what are all the proteins in that? I do a mass spec, I get all the peptide signatures. You can only trace those signatures back to something you understand about half the time, right? Which implies that, either there's a lot of measurement error coming off the instruments, which is certainly true, but there's just also a lot of biology we don't have a handle on yet, which means our measurement instruments aren't sufficient. Okay, I'm going to give you a hint of some of the more radical projects we have underway to give you a sense of what might become possible on the frontiers of biomedicine. This movie in the middle that's just running on loop is bacterial growth on a two-dimensional agarose pad. Cells growing and dividing and growing and dividing. This is what we're familiar with in life. But if you look at this really carefully, you'll see something different happening as well. See those little spheres hanging off the sides of the growing cells? I first learned about these as a student here. They're called mini cells. During otherwise normal growth and division of a bacterium, a single gene mutant will cause a little bleb of cytoplasm to bud off and be encompassed in membrane. An electron micrograph is shown in the right middle. These things have no chromosome, but they do have a capacity to read out DNA and make, of order 100,000 proteins. No genome. They don't grow and divide. They're not part of a replicating lineage. What DNA would you put back into that to boot it up and get it growing and dividing again? How could you escape the world we're in, which is lineage land, where we have all these natural organisms, but we don't perfectly understand them, far from it, and yet we're trying to practice medicine with that, we're trying to practice engineering with that. So the most radical project we've got going on in the lab right now is to create a technocultural framework, and I'll explain the second part of that in a second, that allows people to make lineage-agnostic organisms that are not constrained by natural genomes in an open-ended generative way with the goal of producing organisms for which we know what all the things inside the organism are, such that we might ultimately get to full-on mastery in terms of being able to just simply document everything inside the darn thing. What we're shipping right now is what we call a developer kit to allow people to prototype the different essential modules needed to contribute back into a collective set of potential organisms, right? So we're trying to crowdsource something on a massive scale, massive in terms of technical integration and forcing reuse of materials, but also massive in terms of a global community collectively developing a genome scale object. Thank you for your patience as I don't talk about this very much because it's still in progress. Done? 2 more minutes. I'll just acknowledge a couple things in 2 minutes. My mom came out when we had our second boy, who's now 7 months old, and she and our older boy ate all our chocolate chips. And so when she got back to New London, she mailed us a bag of chocolate chips from King Arthur. It's interesting, on the back of this bag, it says, produced with genetic engineering. We don't have that labeling in California, but you have it here. I think of this as Intel inside, because I love chocolate chips and I love genetic engineering, and so this is a brand thing. Obviously, other people don't feel the same way. I'll point you to one last thing, which is a course I'm teaching right now in the law school. I'm teaching this with David Graywall, who's a law professor and an economist from Yale, but is in residence at Stanford for the year. And the course is based on a hypothesis. Number one, we actually have the capacities to provision everything humanity needs without trashing the planet. Number two, our cultural and political systems inherit a dependency on gradients of scarcity and are therefore incompatible with such flourishing. Number three, 1 and 2 will not spontaneously and constructively self-reconcile. And if so, what do we do about it? Now, this is a hypothesis, and we have about 25 upper-level law students and PhD students working through it, and we'll see how it goes. I'll just share that with you for now. It looks like the hypothesis is holding water, in which case we may struggle to implement a type of constructive manifesto, if you will. If you want to follow along the readings or argue with us, there's all the course readings, and we would welcome that. Thank you for your patience. The slides are freely available here. I am always grateful for the experience I had as a student at Thayer. It's awesome to see how this place is flourishing. I'll thank my labs both at Stanford and MIT, the Helmsley Trust, the Department of Commerce, and also you for your attention. Yep.
And it was quite shocking, the data. So I'm just wondering, the error rate is roughly 1 part per 10 to the minus 2 to be known overall for translation. Do you think that some of that error is coming from this, or do you think that that's a separate error that just has to do with codon mismatch?
The latter. So number one, I have no idea what's going on. We're merely reporting an observation, and a lot of subsequent work is warranted before you make, or anyone makes, extrapolations or broader conclusions. But I think what we're reporting is not merely error in the process of synthesis. My just so story, if you will, is that there is a shadow proteome that's being expressed at about 1% or less of the primary proteome. Think about it this way. When you're hiring somebody on your team, you have to check for at least 2 things. Number one, will they add value to your team? Will they contribute to the fitness of the organism? Number 2, will they not cause problems? Will they not screw things up, right? And you need to check for both. So when a new protein is coming into a proteome, if it causes a problem, that's really, really bad. So the just so story, again, way ahead of where any evidence is that proteins are always being expressed. Cryptic proteins can always be expressed at very low level. If they're really problematic, they'll be selected against and mutations will accrue. Then if you want to promote them into the primary proteome, a single point mutation in the start codon can drastically increase their translation, and then they have a chance of contributing to fitness. Even more dramatically, you could imagine that the anticodon loop in an initiator tRNA could mutate, and that would simultaneously promote a whole family of proteins from the shadow proteome below 1% to the primary. But again, I have no idea. It's just the hypothesis is motivated by this idea of thinking about how you design an integrated system to function not just on a short time scale, but on an evolutionary time scale. Yeah.
Great. I know you have many facts and gifts from Dartmouth and Thayer, so we want to appreciate you coming by celebrating the 150 years.
Thank you.
So while we're setting up the computer for our next speaker, let me make a quick introduction. Our next speaker is also not new to Thayer School of Engineering, and Sam Truex, and she is a Dartmouth and a Thayer alumni. So Sam serves as Synlogic Chief Operating Officer, and she is the head of the corporate development with responsibility for developing, executing on a strategic plan. Before that, Sam has more than 20 years of biotech experience, most recently from Padlock Therapeutics, and that company was acquired by Bristol Myers Squibb. And prior to that, Sam also spent several years at Biogen and Genzyme. At Biogen, Sam serves as a vice president of corporate development, leading the negotiation multiple licensing. So she's really bringing in very unique industry perspective to our frontier engineering in medicine symposium today. In terms of education background, Sam received a bachelor degree in biology from Dartmouth. She's received a BE in biomedical engineering from Thayer School at Dartmouth, and also MBA from our peer department and school across the street from Tuck School of Business. And she has served on Corporate Collaboration Council advising the MEN program at Thayer, and also Sam is a member of Thayer School Board of Overseers. So with that, let's welcome Sam for her presentation.
I'm really honored to be here today speaking at the school, both because I'm with an esteemed group of other speakers and faculty and members of the audience, which is fun and exciting for me. And let's face it, the things that the prior speakers have spoken about are just really cool, okay? I'm not as eloquent as they are. They're just really cool. So I've had a great time listening to these talks. I also feel very honored to be here because I did spend a lot of time here, 7 years on campus as a student, and a lot of times visiting for various reasons. And because if you were here earlier when Dean Helbley opened up the day, you heard him talk about some couple of alums he was with recently who were in the class of 46. And so they can imagine quite a bit of the 150 years of history, not all of it, of course. But my own father was a 47 at Dartmouth and a class of 48 from Thayer. And so I've heard of a lot of the history of the school and seen a lot of its changes, and I'm very excited about more innovation and expansion to come. And I myself, I'm sad I didn't think of putting it in my presentation, but I have a picture of myself from when I was 16 years old, which is 30 years ago now, standing on the front steps during my first visit to Thayer School of Engineering. And those front steps are long gone. Those are 2 iterations of expansion ago. I think they're probably under what was the entryway that's under the Jackson Conference Room. But it's really fun and exciting to have seen and lived through much of the innovation of the school itself and be a part of what's to come as well. So I'm going to offer an industry perspective, and one of the pieces of that perspective is that when you're in a small biotech company and a big biopharma company or a big potential investor says they can meet with you at 3 o'clock, even though that's during your talk at Thayer School, you actually have to be there. So I'm going to go pretty quickly, and I apologize if there isn't a ton of time for questions at the end because I'm literally going to run out of the room to go get on a call. I'm going to offer some of these topics today, but I'd be glad to talk about other industry perspectives. And of course, the folks who spoke to you today, as you could tell from their talks, have a lot of industry experience as well. So I'm going to go through just a few examples of the array of technologies that involve engineering in the medical field, but you've heard of many of those, probably that are more exciting than the ones I'm going to mention today. Then I'm going to particularly talk about engineering and drug development because that's where I've spent my career, and a little bit about going from concept to company. As you heard in the intro, while I worked in big biopharma companies for most of my career, I have spent the last few years in small startup companies, and even while I was in big biopharma, I spent most of my time looking at and evaluating, either for collaboration or acquisition, smaller companies. So I'll give some of the perspectives on that front. So there's a huge array of medical technologies, but I like to start with one that I learned about when I was here in a course that was taught by John Collier, who's still here, and also talk about one that's more recent, the Surgical Innovation Center that some of you may have seen over at the medical school that was in large part driven by another professor who I had when I was here, Keith Paulson. And I thank them for encouraging me to bring engineering together with biology, which I did not know one could do when I first got here. There are a number of other innovations that I think are exciting, and these go along with many that you heard about today that are they're a little more up to date, but perhaps don't seem as innovative as the ones we just heard about. Google Glass, 3D printing, MC10, which you heard about from Dr. Rogers, innovations in in artificial limbs, innovations that bring us the ingenuity of folks like Stephen Hawking. And now I'd like to talk a little bit about engineering and drug development itself. So many of you may know that the engineering of monoclonal antibodies by a company called Adimab came from technology out of the Thayer School of Engineering in Tilman-Gerngross. And there are many other ways that engineering plays a role in drug development, again, some of which you've heard about today. I just looked for some recent developments, nanorobots. There's a whole industry around gene therapy. There's an industry around gene editing that you heard a little bit about earlier from Dr. Cam Leong when he talked about CRISPR-Cas9. These are 3 of the companies, and there are others as well that have arisen based on that technology. A company that I'm now working at actually is a great follow-on to the talk we just heard from Dr. Drew Endy, and that we at this company I've just joined in December, Synlogic, are putting together synthetic biology with the safety and wide use and understanding of E. coli Nissl, which is a non-pathogenic version of E. coli that is used widely in the world as a probiotic. And what Synlogic is doing is actually engineering E. coli Nissl to become an oral therapeutic that will perform metabolic functions that may be missing in the case of various diseases, or to synthesize substances that could be helpful in other cases of disease. I'm not going to spend a lot of time on Synlogic, but I did just, as a plug, add the BioBricks logo because I knew I was going to be following on Dr. Endy, and he talked even much more about some of the things he's done and how his influence is impacting tons and lots of people who will go out in the world and hopefully end up creating more innovation. The background of this company also has a link to Dr. Endy, and that the founding scientists are from MIT, and he actually even mentioned Tim Liu, I believe, as one of the collaborators he has. And I'm going to come back to one of the topics on here, which is there are several great investors in this company, but in addition, there's an enormously experienced management team at this company, and that has developed just over the last year. Even though the company's been around for 3 years, it's really built up on the level of industry experience, and I'm going to come back to that at the very end of the talk. Also in engineering, from a drug development perspective, there are a number of areas that engineering comes into play besides the drug itself. And so this is just an example, and I apologize, I forgot to put the source here. This graphic was given to me by another Thayer School graduate named Amber Bryant, who worked at Genzyme for a number of years and is now at Verily, which is a Google originated company. And what you can see here is the various innovations in actually delivering drugs over the years in the first panel. And then in the second panel, the innovations from a software perspective in monitoring patients and the experience the patients have. Some more examples of that are on this page. This is taken from some information from a friend of mine and former colleague at Biogen, which happens to focus on multiple sclerosis and other neurologic diseases. Just talking about in that particular therapeutic area, various wearable devices that are being used to track patients and how they respond in the case of their disease progression, but also those can be used to help in drug development and understanding how the effect of the drug can be measured. And so this is an area of research that's been going on for a few years and perhaps will even turn into itself a commercializable product. There's some other areas of technology. An example similar to what I said on the last slide is the one on the top, the technology for sensing drug abuse. Another technology, which is interesting, and I'll editorialize a little bit, when I was at Biogen, we spent a lot of time looking at this company, Proteus, which makes the point that there are many patients who need to take drugs chronically and actually are not compliant and don't take them. And so there are a lot of medicines that are sitting on the shelf never taken, which is not only not great for the patient, but also a waste of our healthcare dollars. And so they came up with a technology that essentially involves adding to whatever the oral drug is, a radio sensing technology that would actually be ingested and then a wearable device. My editorial, I hope nobody works for Proteus, is I'm not sure any patient is going to want to do that. You know, it seems a little big brother-ish to have your doctor monitoring whether you took your medicine or not. Presumably, if you could could go to the trouble of wearing the device and sending the data to your doctor, you could have remembered to take the drug in the first place. So I'm not sure, how successful that technology is going to be. But as I was listening to the other innovations being talked about today, I thought about whether there are other, maybe better, more practical ways to improve compliance. In addition to the actual taking and monitoring of the drug, we have the actual manufacturing, formulation, and packaging of the drug, and there's a lot of engineering in this arena. So these are just some pictures. Here's a bioreactor, which I'm sure many of you are familiar with. There's a lot of engineering that goes into the actual filling line. So this is for a liquid therapeutic. This is a lyophilization process. And here we have a bubble pack station. And this may not really seem like huge innovation in the area of engineering, but I have to tell you, having lived through being the program executive for 3 different drugs at Biogen, there are a lot of times where something goes wrong in one of the areas represented by these pictures that cause delays in getting therapies to patients. And so having good engineers and people who are really focused on quality and efficiency in these areas of technology is really important and should not be forgotten. So now I'm going to move into a little bit of a different topic, which is the beginning of a company. And I have to say, while I have been in this industry for over 20 years, I'm pretty new at actually working in a startup. So I'll provide my own views, but to capture some words that Dr. Endy used a few minutes ago, we don't really know what we're doing. So I don't really know what I'm doing, but I'm trying to use everything I've learned over the years to do my best at it. And in the last couple of years, in addition to working in a biotech company, I have been an entrepreneur in residence at a venture firm called Atlas Venture, which is one of the many very productive and esteemed venture companies in the Cambridge, Massachusetts area. And so I've learned a little bit from hanging out with those people, to be honest with you. So let me just go through a couple of things. There are often innovations that come from academia, and in fact, at least in the case of Atlas Venture and many other firms I spoke to over the last year when I was looking at where to go next after Padlock Therapeutics, which I last worked at, we sold that to Bristol Myers Squibb. By the way, there was another Dartmouth 92 on the other side of that deal, which was kind of cool for me. And the head of chemistry, discovery chemistry at Bristol Myers Squibb, also a Dartmouth 92. But he graduated in 91, so I'm pretty sure I never took any chemistry with him because I'm sure he was way ahead of where I was. But anyway, we had 3 Dartmouth 92s sitting around the table at the closing dinner, so that was pretty cool. So many of the people in life sciences venture capital are actually PhDs, MDs, MD, PhDs who have a lot of technical background and sense for what good innovations are. And so not only are they, in the old school way, looking at ideas that are proactively being brought to them, but they're literally going out to academia, listening to talks like we heard today, and thinking to themselves, does that equal a company? And how could I make that into a company and work with that academic founder? And perhaps some of our speakers today and some of you in the audience have experienced that already. And so how are these things funded? There are lots of ways that you can get things funded: grants, angel funding, venture funding, biopharma investment. And there are pros and cons to all of those, and that could be the subject of its own talk, which I won't necessarily go into. But I will just mention that over the years, there has been quite a bit of venture funding available, at least in the biopharma space. And while this is a little bit of an eye chart because I pasted it in so small, this is showing over the course of the last several years the level of investment and the number of investments in biopharma from the venture world. And you can see these data actually end in 2015, so there was quite a spike in 2015, which will be a little bit of a theme for the next couple slides. But in general, there have been funds available, and it has gone up and down a bit, but it's generally been reasonably stable over the years in terms of the ability to get funding for good ideas. And I'll come back to that in a minute. And then building teams is very important in terms of concept to company. I'm going to come back to this in my very last slide where I talk about what you can do as entrepreneurs. There are academic founders, of course, in many cases. Some of those founders are very involved and become part of the company. Some of them are not involved at all after a handoff. And then there are a lot of variations along that spectrum. There are generally people who have been involved in drug development before. There are a lot of functions involved in drug development besides discovery research. Too many to even name here, and to be honest with you, even 15 or 20 years into my career, I was still learning about different functions in the company, Biogen and Genzyme, that I didn't even know existed. So it's pretty complex to get a drug from discovery to being widely available in the world in a safe and in a safe way and a way that's paid for. There's a lot of complexity around that most people don't fully understand. And then it's really important to think about leadership in companies as well. And that again is a topic that could be its own whole talk. But I encourage those of you who are thinking about entrepreneurship not only to think about the innovation in the business model, but how to lead other people who are going to become part of that company. So let me go into a little bit of data first. This was provided by Dan Dubin, who's an investment banker at Leerink. He's also an MD. He's also a graduate of Dartmouth, which is how I convinced him to give me the PowerPoint version of his slide. So I'm going to talk just for a minute about returns on investments. And the reason I'm going to do that is because when you have an innovation that you want to get funding for, the people who are thinking about funding you are always thinking about, and how am I going to get a return on that? And so over the past few years, especially in 2013 and 14, there was quite a boom in terms of returns on biopharma investment. And so what we're showing here is in the bar chart, the actual percentage returns, and then over on the right-hand side, the number of IPOs. So this is initial public offerings for those of you who aren't familiar. So that's when a private company actually offers its stock on a public market. And a lot of times people think, oh, that's the exit for the venture investors. That's the way that everybody who works for the company and all the investors are going to make their money. And increasingly, that's not true. Increasingly, the IPO is just a funding event where more money is put into the company, but that doesn't necessarily mean thereafter it's a liquid stock trading the way that Google or Apple might be trading. And so it often is just an opportunity to get to a place where there's a public currency, but it's not necessarily an exit, especially in the case of drug development, which is a very long cycle to get to being a profitable company. Usually when drug development companies go public, they're not profitable then, and they're not going to be for years. So it's a specialized kind of investor who understands that and has patience for that. And so what you can also see here is that after 2013 and 2014, the number of IPOs dropped precipitously and the return on those dropped, and not because of any fundamental shift in the value proposition of biotech drugs, but because of the perception of volatility in the market, because of the general setting from a governance perspective in our country, and from some pressures on potential pricing of drugs and perception of how that might change with a new president of the United States, which I'll get into a little bit more. So an IPO is a possible way to lead to value creation for an investor. Another way is an exit via acquisition. I just described Padlock having been acquired by the folks at Bristol Myers Squibb. And so M&A, mergers and acquisitions, like I said, is a potential mechanism for exit. And you can see on the left-hand panel, this is the quartiles of the return. So in the red, where you see the numbers with an X after them, that's the fold return over the investment, presumably by venture investors, that was made in some of these companies. And the blue line is showing you the amount invested. So as you can imagine, the more that's invested, the harder it is to get a multifold return. And on the other side, you can see sort of the distribution of outcomes. And remember, these are of companies that got acquired. So this doesn't really include complete failures of companies, because those probably wouldn't be acquired. And even amongst those that got acquired, so presumably have some level of technical success, they're not all these massive exits. And so the folks who invest have to make up for those that lose money and those that only have, say, a 1 or 2 times return, with some that have a much bigger return. And so when they do their diligence on what to invest in, they really have to think a lot about the long-term and short-term risks of the company, which I'll talk about in a minute. And so venture investors, I was discussing a little bit earlier with Dr. Rogers, sometimes although they're very bright and they want to see technologies succeed just as much as the innovators and the management teams do, they at times have a little bit of a different set of incentives than those who want to grow the company for the long term, because they need to provide an exit for their investors in their fund. And they're looking to have one of these or an IPO that at least leads to an exit at some point. Usually within a 10-year window of their fund. And drug development, on average, takes almost 10 years, depending on the drug. So it's an interesting dynamic, the relationship between the leadership team that wants to grow a company for the long term and the venture investors that need to create a return for their investors. So let me give you just a little bit of perspective on the risks, and I'm going to go through some slides that may seem kind of like gloom and doom, but they are a little bit of the reality of this industry, and then I'll at least touch on some great successes. So you can see on the left side, one of the areas of potential value that is partly driven by financial returns, but partly just driven by why do we get up and do what we do in this industry, is because there are patients out there. Some of us may be some of them today, or we might be someday, or our parents are, or our children are. And I'd like to think that while most of what I'm going to talk about involves financial returns and the realities of risks in getting to those returns, what we're really here for is patience. And so hopefully what we look to when we're trying to figure out whether there's a practical application for our technologies is whether someone's really going to benefit from it, and then whether we can make money on that benefit as well. So what investors generally are looking for, whether they be venture investors or they be large biopharma companies that might be looking to partner with or acquire smaller entrepreneurial ventures, is there something that's a new product? Is it in a therapeutic area that's important? Is it innovative, meaning it's going to meet an unmet need? Is it an opportunity to fuel the pipeline that I already have if I'm a biopharma company, meaning it fits with what I'm doing already for some reason, and grows the offerings I may already have to patients? And then is there a way to get it paid for in a way that could be profitable, even given all the risks? And that's what I'm going to talk a little bit more about on the right. There are lots of risks, and this is not a full list, but I'll talk a little bit about the risk profile on the next slide. But getting from discovery to the clinic, and the clinic means testing in humans. Everything that comes before that is preclinical, in vitro studies, animal studies. Then when we get to the clinic, we're in humans. There are manufacturing risks, as I alluded to before. There are risks once you get into the clinic, safety risks, efficacy risks. We heard from a previous speaker about the organ on the chip idea and the possibility of trying to reduce the risk earlier in the setting of developing a drug so that you don't have to spend a lot of time, money, and the hopes of the patients who are in the clinical trials that they'll benefit from something that may not have ever had a chance of succeeding in any event. There are regulatory risks. There's the FDA in the United States and its analogous counterparts all over the world. And by the way, not only are there risks at how they're going to react to any particular therapy, but sometimes the way a regulatory authority in one country behaves and the requirements it has for your drug development are not only inconsistent with what another regulatory agency in another part of the world has, but at worst they can be incompatible. I was in charge of a drug once where one country wanted us to run a clinical trial in a particular way, and another country wanted it run in a different way, and you couldn't, it was a null set. You couldn't run the trial in a way that would satisfy them both, which is really inefficient to have to run two different clinical trials and enroll people from two different parts of the world separately and still power two different trials to show statistical significance, but not be able to use the one trial across the world to do that, especially in a patient population that's rare. So that's something that you really aren't thinking about when you're at the beginning of discovering a drug, but it can become an unfortunate reality. And there's not a lot of negotiating leverage with regulatory authorities. There's intellectual property coverage and freedom to operate. We've talked a lot today about various innovations. One of the things that I don't think was mentioned when Dr. Cam Leong gave his presentation, he talked a lot about CRISPR-Cas9. Just yesterday, there was an announcement of the outcome of an interference case, which is essentially a patent battle between the folks at MIT, the Broad Institute, or I guess it's Harvard and the Broad Institute in Cambridge, Massachusetts, and UC Berkeley in California, that amounts to tens of millions of dollars. And in fact, the market capitalization of two of the companies whose logos I showed earlier went down about 11% yesterday because the patent battle didn't come out quite the way they expected. So these become very important issues when technologies are hot and being used widely. The reimbursement issue is one that it could itself be a whole talk, and it differs quite drastically around the world. Fortunately for us, the United States and Japan as well have been countries that have been really willing to pay generally reasonably high cost for drugs. And while there's a lot of complaint about that, and there's a lot of political pressure on that, which I'll also talk a little bit more in a few minutes about, if it wasn't for that, there wouldn't be drug innovation in the world. We are funding most of the innovation that happens for the benefit of the entire world. Most other countries have a lot of downward pressure on the price of drugs, and while the marginal cost of those particular drugs may be covered by the amounts that are being reimbursed, the funding of all the innovation and all the failures that happen along the way, if we go too far with pressure down on drug pricing, is not going to be covered by the amounts that are being reimbursed. And we're going to end up in a situation where we don't have much innovation anymore in drug development. And so that's something to think about when you listen to debates about drug pricing. And then there's always commercial risk, of course, which I won't dwell on a lot. That's probably pretty obvious. But sometimes, as we discussed a little bit at lunch, sometimes one of the hardest things to do is kill a project when it isn't really working or it isn't really living up to the innovative potential one thought it should. And developing a drug and getting it approved is one thing, but you can get a drug approved that's not any better than something that's already out there. The FDA is there to make sure your drug is safe. It's not there to make sure that your drug is better than what's already available. And so one of the challenges I've seen and lived through at big biopharma companies is sometimes there are pet projects and there are drugs that get approved, but they're kind of ho-hum in their commercial achievement, and that's because they're not really all that much better than something that was already available. So let me take a quick look at this picture, which is not too different from some of the points you already heard made earlier today by Dr. Cam Leong, if you were here. This is just a picture from a Pharma's an Industry organization, and in their annual report, and that you can find these numbers in lots of different sources, and they're slightly different. But the point is, if you start at discovery with thousands of compounds, the numbers that actually ever get into clinical trials, which again means testing in humans, are pretty small. And from that, here they're showing 5 gets to 1 FDA approved. And I've seen other predictions that say it's as stark as 10 to 1, so 10% chance of approval. And earlier we even heard, I think, 5 or 6% in I think it was oncology and neuropsychiatry in the slides we saw earlier today. So this is a risky business, and most things fail. Okay? That's the reality. Hate to say it, kind of stark. But some of them succeed, and that's why we keep doing what we're doing. Otherwise, you'd think it was futile, and why wouldn't you go, like, open up a bakery or something? Because generally you can get the bread to be made, I think. I don't cook, but... and in addition to it being very risky, it's very expensive, and we heard a little bit about this earlier. So this is, again, you can find different sources of data with different numbers, but this is showing over the course of a 10-year period, which is now a few years old, over various companies, Biogen, Celgene, Gilead, Amgen, what is the cost per launch of their drug, not only for the research and development, but also the acquisition cost. And so sometimes those innovations were acquired at some earlier stage of their development. I talked earlier about M&A and all the big exits. Most of those M&A deals were not done when the drug was already approved. Most of them were done at some point before it got approved, and large dollars, as you saw, were paid for them. So this is the expense that these companies have had in the billions of dollars have had to spend for the launches of each drug over those years. So why do people do it? So I talked about venture investors needing to have an exit, and most of the exits, as I said, meaning that the time when they can sell the shares they had in a company, happened before the regulatory approval, as I just described. And so you might say, well, why do large biopharma companies do it? How can they possibly invest in anything when any one project is more than not likely to fail? And the answer really is that they have a basket of risk, right? So they have a whole pipeline of drugs, and they try lots of shots on goal, and some of them succeed. And we all benefit from that. Society benefits. Hopefully the people who have the diseases these drugs treat are benefiting. And even those that fail, hopefully there's enough publication and sharing of the data about those failures that the next iteration can benefit from those. And I'll talk about an example of that in a minute. I already mentioned that the US and Japan generally have higher drug prices. I also said there's been some pressure on that. So here's just a little bit of an older set of data, but suggesting that pricing pressures began years ago when generic competition, which has been present for decades, but now is becoming more and more of a pressing concern, particularly as biologics. And you saw earlier, I think, from Dr. Endy, how much money is in our GDP in the case of biologics, which are therapeutics that are produced as biologics. And so here you can see in the last 5 years or so, $93 billion in U.S. sales was exposed to generic competition. When you have a small molecule that's synthesized and it, let's say on average it's at a price that's scaled to 100, after a generic competitor comes in, the price drops to about 20. So that's great for those who are paying for it. That's fantastic. But for the people who spent these billions of dollars developing that drug, they have a real issue. They have to make sure they've made up for the expense that it took to develop that drug before that generic competition enters. And this is why patents are so important, and the life of patents is very important. The numbers I just gave, the numbers I just gave were based on small molecules. For biologics, the price doesn't fall quite that much because it's a lot more expensive to develop a biologic. So biosimilars don't have quite as big of a cliff that they fall off in terms of profitability when loss of patent exclusivity happens. But if you hear debate in the general government about drug pricing and how that relates to patent term, this is why. So let me also go to, in the news, especially in the last year, there have been a lot of headlines that relate to drug pricing. And some of them, honestly, again, I'll editorialize, I am just mortified by and don't want to be associated with at all. So here's an example. Drug goes from $13.50 a tablet to $750 overnight. This is the case of this young entrepreneur who decided he saw an opportunity to make money by finding a drug that was very low price, kind of old, but only one company was making it because it wasn't very profitable, not sexy anymore, not a big deal. But patients with cancer and various other diseases who could be susceptible to life-threatening infections rely on this drug. So this guy buys this company and jacks up the price from $13.50 to $750 with no innovation, no reason for jacking up the price other than I can. So there is some skepticism about the industry, there is some criticism of the industry, and this is a reason why that's warranted. I find this reprehensible, and I think most people in the industry do, and if you have any interest, you can look up a lot of vitriol aimed toward the fellow who was responsible for leading a company this way. He's no longer there, by the way. Now, there was pricing pressure that some people who don't understand the industry might put right in the same category from a company called Gilead, which brought up a hepatitis C drug and charged something like $83,000 a year for the therapy. That may seem like a lot of money. There are a lot of drugs that cost in the tens of thousands of dollars a year for the therapy, and it's partly for the reason I described before. It costs billions of dollars to develop the drug. This drug cures hepatitis C, a chronic, debilitating disease that can lead to death, liver transplant, etc. This I'm proud of. This resulted in the government laying out for public view how this company decided to price at that level, and they made this company, Gilead, look like bad guys for trying to figure out what was the highest price they could go to without causing a backlash. And they made that sound like an evil proposition. Guess what? That's how all drug companies try to figure out how to price their drugs when they launch a new drug. So this one, I think, is a tale of how drug companies should behave, but a lot of people in the media or who don't understand this industry kind of equated those two as both bad guys, and I think that's too bad. Another example of what I think is a bad guy is Mylan's EpiPen pricing, where they, and this was also in the news last year, decided that an EpiPen, which again is kind of a commodity, not all that innovative, but they decided it should cost $600. So we could go on, and this guy probably is going to go on, and described in January of this year, during one of the biggest biopharma conferences of the year in San Francisco, how everybody in pharma is getting away with murder. Well, like I said, some of the examples here may cause us to say, well, we deserve that comment, and maybe not murder. And some of the things that this industry is doing are really no different than any other industry, like Microsoft or Tesla are doing when they decide the prices of their innovations. So there are some things to celebrate, and in this slide, one of my pet peeves is too many words on a slide, so I didn't do a good job of this one, and I won't read it all to you. But this is the example I just gave of Gilead's drug for hepatitis C. Devastating disease. The way it was treated before was difficult, if not horrible, and now a once-daily single-tablet regimen offers patients, not everyone gets cured, but a very high cure rate. Now, for full disclosure, these words are actually from Gilead's own materials, but I know enough about this drug, and by the way, Gilead's materials have been scrutinized, every single word of them, by the FDA, because that's what happens in this industry. You can't make any claims about your drugs without every word of that claim being reviewed by the FDA to assure what you're saying is substantiated by data. This is something to celebrate. Another example is immuno-oncology, and there are many examples of this, but the most prominent are anti-PD-1 antibodies. Both Merck and Bristol-Myers Squibb have these. And I would point out that when I worked at Genzyme in the year 2000, 2001, I was working on ways to harness the immune system to fight cancer, and so were about 10 other companies. All those companies failed. And companies before them failed, and companies after them failed. But everything they learned was built on, and stacked on the shoulders of all those failures were people who did learn ways to harness the immune system and finally succeeded. And so one of the big beneficiaries who was in the news is Jimmy Carter, who took one of these drugs. I believe it was, well, it doesn't even matter whose drug it was because they're basically the same, but was able to fight a cancer that seemed like it was going to be devastating just by using a therapy that allows your immune system to rev up and fight your own cancer. So last thing that I wanted to just quickly point out is what can you do if you're an entrepreneur? And there could be 45 slides on this, but just a few things I want to say are, be clear on what your data are and the implications of that. Be able to communicate that clearly and really effectively. Be transparent about what your patent situation is and pursue patents. Know that you don't know everything. This goes back to when I was talking about Synlogic hiring lots of people. There's over 175 years of experience in the industry across the management team of that company, and that's not unusual in these companies. Like I said, there's a lot goes into getting a drug to patients safely and effectively and getting it paid for, and no one entrepreneur can do all of that. So find people who know the path and listen to them. That doesn't mean don't question them. It just means learn from them. And don't think you can do it all yourself, because that's a naive view, and I have seen entrepreneurs who take that view and they don't generally do very well. And then lastly, balance the sell with the current reality. So being on the buy side of business development for many years, I saw a lot of situations where companies would come to us at Biogen or Genzyme and talk about what their vision was as if they had already achieved it, and try to sell something that was where they were going to be in about 5 years. They may have thought they were going to be there in 5 months, but usually that's not the case. So it's great to have a vision and articulate it, but then be candid about where you are, what you need to get there, and how whoever you're talking to can help you get there, either with funding or their expertise. So I'm going to stop there and maybe take, I don't know, a question. Thanks very much. Well, everybody does. I don't mean to. Just mine happens to be in 5 minutes.
So Sam gonna leave in 5 minutes for a coffee phone call, but now we can open for maybe one question from the audience. Yeah.
If Trump really doing some integrated of the drug, like the FDA approval process, what do you think will affect the entrepreneurship of the drug development?
That's a really interesting question. And yesterday I saw a headline, and I follow a number of blogs and sort of the Twitter sphere, if you will, of the Cambridge Mass biotech industry. I haven't seen any commentary from any experienced drug developer that says they are in favor of making the FDA less restrictive. So I don't think it's actually going to happen, because I don't think anybody thinks it's actually responsible. Although, to be fair, I don't think we know exactly what he's talking about. I mean, I'm not going to, I don't think we know crisply what changes he's talking about making. There could be some that could make the FDA more streamlined, and that would be great. But not regulating and reviewing the safety of the FDA is not a good idea in any drug developer's mind. And so, again, my answer is I don't think it's going to change entrepreneurship, because I don't think it's really going to happen.
All right, Sam.
Thank you so much.
And appreciate